Here we are. And at the end of class today, we're going to have a drawing assignment. It's going to be based on the clay model videos, so it's a review. It should be stuff we're really solid on. And we just want to conceptualize another step up in understanding and appreciating how we're using that clay model video, okay? And so we'll do that. And because of that, we won't have a concept chat today. We'll just roll into that. And we'll do that, with about 20 minutes left, and we'll kind of zip along through it because it's largely a review. But it's also this intention to demonstrate the value of drawing this stuff out. Like for reviewing, you're building, your study guides and stuff like that as you're learning. That's what we're going to do. All right, so we'll do that today, and we won't be doing that one that we lost during the snow day. Then we have Riley, and we know Riley, and you can ask questions about that. Riley's going to be here for a total of 4 times, but Riley's always available as a course tutor, and that's really important because the Learning Center can help with tutoring topics like study skills and stuff like that, but really don't have expertise, typically at this level. So he's our direct go-to, and you can contact him. One thing that's a little bit weird is to send an email to our student and say, oh, can we meet by Zoom? I really like the idea of teaming up, with a classmate, anytime. Talking to me, right? Come to meet with me. I just think that's intimidating. I think I mentioned this before. Come with a classmate or two, and meet together. I think that's a really nice thing to think about because I do think it is kind of spooky to reach out to Riley or me, like that type of thing. And I want to be really supportive of that. So where we're at is we're going to look at, I want to start reading chapter 9 and this week. And chapter 9 is about transcription regulation. Our book doesn't approach it by trying to lay it out like a clay model video, but that's what the book is doing. So we're going to take that understanding and we'll add to it, and we'll have questions about it because the book covers more territory than we do. And but we'll get to the important parts that we want to add to in class probably by the end of the day or certainly by Thursday. So that's what we're rolling forward on. We're still talking about chapter 4. That was an intro to the fundamentals of molecular biology. And you can see, well, I haven't said hardly anything directly about chapter 4, but we have examples to go to, like the clay model videos and the signal transduction pathways, anytime we want to talk about the fundamentals of molecular biology. And so we want to bring questions to class like that, because you'll see that's like that our lectures will become more similar to what's covered in the chapters, but they're always going to be different because I think the author is great. This is the 8th edition of this book. He's been writing about this stuff his whole adult life, he's doing a good job. So you should be able to understand that stuff, and I should be able to ask you the good, questions out of it. Not trick questions, not trivial questions, but, what are the important questions that are being dealt with there. So it's good challenge for us as biologists in training to tackle, reading and biology. Just to look ahead, after chapter 9, we go through section 4, and this is what section 4 looks like: cell signaling. So that's all about that receptor and the signal transduction pathway. So we really have been foreshadowing this chapter from day one. It's one of our pivotal chapters. Then after that, it's about the cell cycle, and that's the regulation of cell growth in our bodies and other eukaryotes like that, plants and animals, and single-celled eukaryotes like the yeast that cause pressurized. And that's a really good one to think of for us. We've already talked about as a cell response. So that's what's happening in the skin cells under a wound. Is PGF is binding the PGF receptor, and we have this signal transduction cascade to activate ELK, and ELK is going to activate transcription of cyclins, and then cyclins play a big role in the story of the cell cycle. So we're also kind of pointing toward that. And then it turns out we saw just briefly that when you activate PGF receptor, it's not just the RAS, RAF, MEK, or ELK pathway. It activates several different pathways. And one of the things that happens a lot when a cell is stimulated to grow in our bodies is it starts to think it should kill itself. Like that is interpreted to be a natural mechanism to avoid cancer, is to have this controlled growth, and nobody's going to get out of line, nobody's going to get going too fast, and if you do, you should just go through this process called programmed cell death, like that. So that chapter 19, that's why it comes up. It comes up because it turns out for cancer, a lot of times, there are mutations in growth pathways, chapter 17, like pathways, and, or 18 pathways, and also that cancers tend to have mutations where some cancers have a decrease in their willingness to go through the programmed cell death. And we'll see some really famous examples of that, where the cells just won't kill themselves. They won't, they won't fix damaged DNA. They'll just keep rolling forward like that. So that's why that fits in there. And then chapter 20 is really a cancer biology. I think it might be called cancer, and but it just stays on that molecular biology level of molecular mechanisms. And we will see that some of the stuff we've already talked about isn't really dealt with in the book till chapter 20. For example, chapter 20 has a section, just one section, one figure on CAR T cell therapy, like that. So these come together. How far will we get before our first exam? Well, maybe through chapter 18, I think, like that, probably like that. So, our first exam will cover everything up to that date, basically, and it'll be the videos and the handouts and the assignments and lecture class meetings and the book readings, like that, okay? So that's where we're at, and just one more slide about this background. There's Riley's email address and stuff like that, and just, writing out this idea of how we can get learning support for this class. Okay, so let's just start here and with any questions people have about anything at all. Yep?
Is there a date for the first exam?
There is. It's on our syllabus. It's not for a while. It's not for like another month, I think. I think it's March 6th, but it's on our syllabus. And all of our exams will have 3 exams, and they'll all have 50 short answer questions, each worth 2 points. So that's the kind of learning we're doing, for our course, is this boom, boom, boom. Like, similar to our quiz, students in the past have said, oh, the exam was harder than the quiz. I think that's mostly because there's just a lot of material to learn, like that. And the quiz was just focusing on those videos for the muscle development.
Do we have another quiz?
Yeah, that's a good question. I don't think we'll have another quiz. I think we'll have more assignments. Yeah. Other questions people have about anything at all? Remember that first time? Yeah.
Go through that in chapter 4, like the diaglot, so anyone should go through those things?
I will if you want. So you should read it, and if you have questions. But like, are you asking a question right now, Godwin, about something?
No, because I was just reading it.
Yeah, yeah, and this is a 400 level class, so it's kind of a class where I'm not going to go through, here's the next slide, next PowerPoint in the chapter, here's the next, like that. And they'll become more similar, but to start our course, they're really kind of different and complementary in how the author and we want to get there. And so it's super important. Always remember the first time I taught this class, I came in and I said, I don't have anything to say. It was a different textbook. It was more similar to the kind of stuff I talk about. And, I'm just like, gosh, I mean, every word in there is just like how I talk, because it was 6 of the 8 authors were people I knew and had in grad school and stuff. I'm just like, we're on the same team. This author's on the same team, but has a different angle like that. And so there's a lot of stuff to add to it. Yeah. But any other questions for now? And remember, you can just interrupt. Like, you don't have to sit down in class and be looking at your questions. You can just come up with questions. It's my job to, field whatever question is and fit it in, like that. Yep. So don't hesitate to ask questions. That first time I taught it, there was a student, I can't remember his name right now, but he asked a lot of questions. And then after that course, he took the MCAT and he got 1 wrong on the MCAT. And that happens across the United States maybe once or twice every year. Nobody gets them all right on the MCAT, and nobody gets them 1 wrong. But that student was just asking questions, and a lot of this stuff is, I'm not teaching an MCAT prep course or a medicine prep course. It's true for any animal. It's all true for any animal, and it's got really broader stuff for all life on Earth, like that, But oh my gosh, when you're talking about cancer biology, they want to ask you about that on the MCAT. And see if you can understand modern medicine at all, like that. So anyway, so questions. Interrupt anytime. Don't hesitate. One of the reasons we do that chat is to try to get, open things up and get you, kind of ready to ask questions, stuff like that. Okay, so that's Riley's stuff, and we talked about it some last time, and it's an interesting thing, right? Because Riley's covering different things, like really issues with medicine and providing medicine and cost of medicine, stuff like that, right? It's all relevant. In our course, it's just like, okay, so we're going to build this modified CAR, chimeric antigen receptor, and we're going to do it by manipulating the coding regions of the DNA for 3 different genes in a test tube and build it. Then we got to get into these T cells. We got to get them out of the patient, rewrap a virus around that DNA we made, and get it into the cells and put them back in this organism. So we're really interested in that. Just fascinated by that because it really helps us understand the principles of gene organization and receptor biology. Okay? And then we're just using this. We'll use this today like that, and we'll have this drawing thing. Look at all these beautiful markers I have, and this drawing assignment, and we'll roll through that right at the last part of our course. And we'll really roll through it because it's familiar territory, but I want to demonstrate that it's good to draw it out, and then I also want to add some to it. Because, I don't want to name names, but like I got biology professors, like they, biology professor anyway, that professor, and some people, they just have a different way of expressing stuff, and I'm like, I'm just be trained to just be like, yeah, no, like that, it's amazing like that, to express this like that. Like this statement, the only output from DNA is RNA. I've never heard anybody say that, but I think it's pretty true. I don't know, just like, let's get to the nuts and bolts facts of the situation. All right, so here's our author, and we talked that he has spent his time really studying a natural biological pathway in our bodies, and it's a set of pathways like that, and he's interested in a signaling molecule, a receptor, and this signal transduction pathway and the cellular response. So that's his entire career, that chapter 17. He started out studying a virus that we'll talk about today, and scientists, he and others, found that the problem with that virus is it could cause cancer, and the reason that virus could cause cancer is because it had accidentally, just totally accidentally, picked up a host gene. And it was really the host gene that was defective, that was mutated in some way, that was causing the cancer. And that was discovered, those discoveries were made back in the 1970s, so 50 years ago, like that. But there's still some really famous examples. The gene he studied, RAS, in the 1970s, is found and mutated in 1/3 of all human tumors. Nothing to do with the virus, it's just that they used those examples of those viruses in the 1970s to really got to get their foot in the door, and very early on understanding molecular mechanism of cancer, like that, okay? So he worked in this guy's lab. Again, you don't need to know names, but just it was in Wisconsin, and this guy, Howard Temmon, was really interested in trying to figure out what was the problem with these viruses, okay? So let me just show you this. You don't need to scramble to write it down, but viruses can be organized into some different categories. A little bit like, like we can talk about, there are vertebrates, and with the vertebrates we got mammals and reptiles and amphibians like that. So you can create sort of evolutionary relationships between viruses, and they fit into some different categories like that. But viruses evolve and change really fast, and so these categories are kind of problematic, right? Like that. This category system here is just based on if you get the virus itself. Is it made of DNA or RNA? Is it double-stranded DNA? And which strand gets copied, like that. So it's really kind of a mechanism of the replication of the virus. And the viruses we're interested in fit into this category that is somewhere represented here. It's represented by an example of HIV, and they're called retroviruses. And so we're interested in this category. And HIV is a retrovirus in humans, and HIV doesn't directly cause cancer. What happens in HIV is it infects specific cell types in the immune system that weaken the immune system, and with the weakened immune system, HIV patients can get pneumonia and other infections really easily and get really sick because their immune system is weak, or they could get a cancer and not be able to fight the cancer. So HIV is a good example of a virus that doesn't cause cancer directly. There were some retroviruses that have been discovered sort of back in the 1950s and 60s that could cause cancer directly if you just infected an organism with that cancer. And they weren't humans, they were other species, some in birds and mice and monkey, like that. a couple other species like that. And these viruses were just curious. They were all retroviruses, and they could pick up, they could get to cause infection and cause cancer right away, okay? And this is how they did it. And so this was worked out in some labs, including that lab in Wisconsin where our author, Jeff Cooper, worked. And so we'll need this would be a good 2-point question on the exam, is just draw and label the retrovirus life cycle. So here's the retrovirus. It's outside of the cell that's going to infect, like that. And just imagine that what's one happens to be like it's a mouse, you know, and the mouse gets this virus infection, you know, like you get infection, and the virus is going to enter the host cell and replicate and then make more viruses like that, right? So this is the infection cycle. So here it is, it's RNA in the virus. So we start out as RNA and it infects the cell. And then after infection, it needs to replicate. It needs to make more virus in the infected cell. And the way it does that is it first copies the RNA into DNA. And that process is called, and maybe instead of using this, I'll just draw it, the whole thing over here, because we can show our highlights pretty easily. So here's a cell membrane of the, cell is going to get infected. Here's the virus. And it has an RNA genome. And it's in a category of RNA viruses that are called retroviruses. And retroviruses have the same replication cycle. So it infects the cell and the RNA comes into the cell. And the first thing that happens inside the cell is it gets transcribed into DNA. And that process is the opposite of what's normal. Normally we take DNA and we transcribe it into RNA, like in the clay model videos. This is the opposite, and so this process here is given a special name. It's called reverse transcription. And there's an enzyme that needs to do it, and it's a polymerase enzyme. It's going to grab little DNA letters, A, G, C, T, and going to add them as needed using the RNA as the template. And that little enzyme is called reverse transcriptase. And it's a detail, but the detail is just to say that actually the very first thing that has to happen when the virus infects is that we need to transcribe the gene for the reverse transcriptase, and we need to make the reverse transcriptase enzyme. Because the RNA virus needs to provide that.
So the RNA provides the
reverse transcriptase enzyme, and it does that by having a reverse transcriptase gene, and there's the gene and the RNA, and so the ribosome binds to the RNA and it just translates that gene into reverse transcriptase enzyme.
So both that purple and that green are reverse transcriptase?
Yeah. So you'll see, we'll want to talk about reverse transcription and reverse transcriptase a little bit, because that comes up in chapter 4 as one of the fundamentals of molecular biology. And it's an example where the scientists discovered this and they said, hey, we could use that as a tool. We could make new experimental methods, and that reverse transcriptase enzyme could be used as a tool to help us make more discoveries. Not just about viruses, but for example, in the lab class for our course, we use reverse transcriptase to help us learn about a caterpillar, right? So we use it, take advantage of it. Companies sell that enzyme, we buy it, we use it, like that. So that's why it pops up as like, oh, okay, yeah. And that's this Howard Temin lab in Wisconsin, and he discovered this pathway and got a Nobel Prize because partly because of its help in understanding those cancers that are being studied, and partly because scientists realize, whoa, that's a powerful enzyme. We can use that a lot to help us with our discoveries. Okay, so now we got our DNA, and the DNA goes into the nucleus, and it gets integrated, it gets put into the chromosome. And let's give the chromosome another color. So we're going to have the host chromosome, DNA, and we're going to get that virus DNA inserted right into the host chromosome. And now we can do this regular transcription, like we've learned about that the host cell does. Oops, that's a little bit long, but we're going to transcribe that region and make RNA products. We should have put them in here. So here's RNA. And those can be used to produce more viruses. So that's the replication cycle. Is infection, then reverse transcription, then insertion, then transcription, like that. Okay? Yep.
So does the DNA chromosome transcript to make RNA
Yep.
products?
Yeah. And that goes back to what the host normally does. Like the clay model videos is we've got DNA in the chromosomes, and RNA polymerase is going to transcribe it into RNA. So there the virus is taking advantage of the host cell's molecular machinery to replicate itself a bunch, like that, okay? All right, and so, kind of sloppy drawing on my part, I'm sorry, but that's, what happens, but, those steps. And those are just the key steps to say on an exam, just draw the replication cycle and label the replication cycle of reverse transcription. And you'd be like infection, reverse transcription, insertion of DNA into the chromosome, and transcription and packaging new viruses. Yep.
How does it get added to the DNA? Is there like an enzyme?
Yeah, it gets added to the DNA by an insertion event that's a recombination event that's like DNA repair.
Oh.
Like it's taking advantage of the cells thinks, oh, there's an error in the DNA, I'll fix it, and it does that repair, and in the process does the repair by a recombination
Is it
event.
like an enzyme or something?
Mm-hmm. Yeah, it's a series of enzymes and DNA binding proteins that guide that, because it's mostly that you have these DNA ends, and whenever there are ends of DNA like that, the cell is like, oh, well, a chromosome's been damaged and I need to repair it, like that. So I'm going to bring together the enzymes I need to insert this, glue this in the DNA. And it finds a spot that it's decided that is the spot, and it just cuts the chromosome and inserts it. Yeah.
Can you repeat the steps again?
Yeah. So the steps, because that's what you want to say. Just for this is, what are the steps in the replication cycle of a retrovirus? Any retroviruses, and then it's infection, reverse transcription, then insertion of DNA into the chromosome, then transcription, then like packaging. And retroviruses package, they leave the cell by budding, so the cell doesn't burst. Sometimes you've learned that the cell can just burst and the viruses are released. Retroviruses leave the cell by budding. Yeah, okay, yep.
So, do retroviruses typically not kill the host?
So that's a good question. You would say that they don't kill the host cell like that, but whatever cell they're infecting is really struggling, and that's what happens with HIV. Is that the retroviruses infect a subset of the immune system cells, and they really eliminate those or weaken those to the point of uselessness, and they're critical in our immune response. So in that sense, the virus doesn't care about what's happening to the body very much, right? It's just replicating, but it happens to be replicating in a way that's really problematic for the human body. Yep. Okay, so really the replication cycle, right? And and our author is helping discover that when he's your age. And then this reverse transcriptase enzyme is part of it, and that's something that we'll talk a little bit more about because it's used as a tool like that. We've talked about it some in the lab class already. And then here's the key thing for the cancer story. The key thing for the cancer story is right here, and it's at this site of insertion. The site of insertion is pretty random. So we could just insert that kind of anywhere in the chromosome. And so now I'm just going to erase that, and we're going to zoom in on that, because what happens is viruses get replicated a lot. They make a lot, and so they make a lot of mistakes. There isn't that kind of natural selection pressure to be accurate. So they make lots and lots of mistakes, lots of mutations, and so this is what it looks like. We have and maybe these colors don't match, but there's the host cell's DNA chromosome, and we've inserted this retrovirus genome into the chromosome. And now the cell's own RNA polymerase, like we know transcribes DNA, the RNA polymerase is going to land here, and it's going to copy this like it's a transcription unit. It's just going to land there and transcribe it. But it's sloppy, and sometimes by error, it starts here, and it actually copies out, and it includes part of the host DNA. And so there we get this incorrect RNA that's this long, and it's an error, but it gets packaged and it goes on to infect other cells. And that's a kind of mutation, or it's an error because it's a mutation because we just made a mistake by adding more DNA to the retrovirus than it's supposed to have. Okay? And that's the problem that these scientists were studying with these retroviruses that could cause cancer. Because one of the retroviruses, the one that our authors studied, had accidentally picked up the host DNA that had included the gene for RAS. And now this retrovirus had RAS, and it infected future cells. And then the RAS could pick up a mutation, and it could cause trouble. It could cause cancer. Because we've seen that RAS is on a growth pathway. And if it's defective, it could cause cancer. And so in those retroviruses, and they are referred to all together as oncogenic, that means cancer-causing retroviruses. And these were really stories from the 1970s, but they identified genes that we still study today, RAS. Okay?
So the likelihood of RAS getting mutated is high because the RNA polymerase from the viral genome is sloppy, so it could insert a codon or something like that.
Yep. Yeah. And so when it first picked up the host gene, the host gene might have got damaged just in that process, and we'll talk about examples of that a little bit. But also, it could just be fine. RAS could be fine, and then later it picks up a mutation because it's just being dragged along when the retrovirus, replication cycle, and there's really high rates of mutation errors in retrovirus that's copying that. So it, pretty quickly could pick up a mutation. Then it's forever cancer causing, right?
I guess my question is, why would the viral RNA polymerase be more likely to mutate than the cell's RNA polymerase if it's the same RNA polymerase?
So the key player in this is the reverse transcriptase.
Gotcha.
And so in the normal replication, it's in the host, the replication of the DNA is through DNA polymerase, and there are some error correction mechanisms in the enzyme itself, and reverse transcriptase doesn't have any of that, because you could make 1,000 copies, and if only 800 of them are okay, that's fine. There's just no natural selective pressure to do a really good job like that. So it's just sloppy, and you make a lot. And that's why we get all these variants of COVID, because it's sloppy and you make a lot. It's not like the virus is trying to be mean or stuff. It's just like, it's like somebody that's got to make 1,000 burgers an hour, and pretty soon the burgers are looking pretty crappy, because they're just like, poosh, I don't care. I'm making 800 good ones, and we only get 200 complaints an hour. Fire me. See if you can find anybody else to do this job. So that's what's going on, right? Like that. Okay? So that's the key thing. And there's a set from the 1970s of about 24 genes that were identified. And they were called oncogenes in the 1970s. And in 1971, the federal government declared war on cancer. So in the 1960s, the federal government had poured money into landing people on the moon, and that was successful. And so then they're like, wow, that worked great. Let's do another big, American effort. And so it was called the War on Cancer, and it's still going on, and it's just got tremendous amounts of funding. And the only people that could really help are these people like our author. We're standing there in the 1970s going, oh, we found some oncogenes in the host, okay? Like that. So we'll turn to them and use those as an example a couple of times, like that. Just a couple of times like that. And because the examples are really satisfying, like that. And there are a couple of questions, maybe. There's a question there, yeah?
So the oncogenic, retroviruses, can you just explain that one more time?
Yeah, so there's a category of viruses called retroviruses, and they all go through this same replication cycle, all of them. HIV replicates that way. So there's a bunch of different ones out in nature. Some infect cats and dogs and monkeys and so forth, like birds, like that, right? So it's possible, just by very rare chance out there in the real world, that one of them makes a mistake and picks up some of the host gene information, and sometimes that's a gene that could cause trouble. And so when that virus goes on to infect another cell, that could cause cancer. And it's because of the host gene that's the cancer. The retrovirus does not cause cancer. The host gene causes cancer. Yeah, here's a good way to think about it. So in Wisconsin, our author identified a host gene. And this was way back where they didn't have names for them. That just, there's host gene information there. Like that. It's just very, very long time ago with regard to the tools these scientists had. So then he went to Boston, I think to Harvard. He's going to study it further. And here's what he did. He took some mice in a lab at Harvard that had cancer, and he took out the tumor, and he ground up the cells, he got the DNA, and he tore the DNA into small pieces, and he injected those into healthy mice. And some of those mice got cancers, and he looked, and he found that some of the cancers had that same mouse gene. Nothing to do with the virus. The virus was just the first foot in the door, and then they turned to a regular mouse that just got cancer. And they found that that regular mouse's genes that got cancer were the same DNA, like that, right? So it's important because our author talks about this in the book, and it's just important to appreciate that all the talk of those oncogenic retroviruses is just focused on getting started. In the 1970s, when there was a lot of money to figure out what the heck is going on. And that's the really nice first experiment. Again, you don't have to remember our author's name or the Wisconsin-Boston, but it's just nice to see. He's in Wisconsin, working with Howard Temin, who's just trying to figure out the replication cycle. Discovers reverse transcriptase, wins the Nobel Prize. Discovers that they insert into the DNA, and that just immediately suggests, oh wow, maybe these things have picked up some defective host gene, and it's the host gene that's causing trouble. So then our young author, your age, goes to Boston, and he says, leave the virus stuff in Wisconsin, I'll go to some mice that have cancer. I'll get their DNA. I assume their DNA has some mutation in some important gene that causes cancer. I'll break that DNA up, and I'll just start injecting into mice. And if a mouse gets cancer right away, I'll look, and I'll see which DNA that was. And right away, he found this one, and they call it RAS. It's the RAS gene. It's defective, like that. So it got defective in the retrovirus to start with, but then leave that behind and go to the natural animal, and it got defective, like there. Yep?
So if the virus is infecting this initial host, is the initial host also going to pick up a cancer-related RAS? Or is
No.
it just
So
gonna be
originally, this retrovirus is just going to make the animal sick somehow,
Yep.
and it's fine. But then, by accident, just one animal picks up that, and then that becomes a problem. That's like a variant, like that, of
when
the
it's,
original.
Passed to other
Yeah, but it's
organisms?
passed to other organisms, like that, or like that. Yep. And that's what they found is they have these viruses that they isolated in the 1960s, and those variants of those retroviruses just immediately cause cancer, like within a week. They're like, whoa, look at that tumor growing, like that. Yep. Okay. So RAS could do that, like that. And so then this is not till chapter 20. So some of this stuff in chapter 20. Oh, you had a question a while ago.
So that section of the genome that the virus picks up, does that already have to be mutated to cause cancer in order for the virus to then cause cancer in other organisms, or can it just be any section
Well,
of
I would
the
say probably nobody knows for sure, but the really strong interpretation is no, it's fine. It's fine when it gets picked up, like that.
But the section
Yep.
itself of DNA, does that already have to be the section that it picks up, does that already have to be a cancer-causing section of the genome in order for it to then go on and cause cancer in another organism?
That's a good question, and let's say, then all these questions are good questions. Sometimes I just say that, how, like I say, you say, yeah, good question, but that's a good question. So the interpretation is that in our bodies right now, there are these genes like RAS that are not mutated, and they're doing their job, and their job is in regulating growth. If they get mutated, we'll have a problem with growth that could lead to cancer, like that. And so those genes are called oncogenes, and they're ones that, if mutated, would especially lead to cancer. And so that's the interpretation: is that the virus happened to insert next to an oncogene, and then happened to accidentally take that with them, and now is with the virus, and it could get mutated, and it could cause that problem
Okay.
with the virus story. Yep.
So is there a way to detect oncogene in a person?
Oh yeah. Because nowadays we know a lot of these genes. We've studied them a lot. Scientists have studied them a lot, like that. Yep. But we want to go back to that just because the book is talking about those oncogenes along the way. Those pop up as the most famous oncogenes, like that, and they're historic, but they're still current. I'll just describe another one for now, and you'll be like, okay, all right, like that. So, just to describe another oncogene. So, let me show you. I have a list I made of these ones that were originally isolated, right? There are about 24, I think it's like 30 genes that were identified that were normal host genes, but could get mutated and lead to cancer. And so those genes are called oncogenes, cancer-causing genes, and they're in our body right now, and they're doing their normal job, and that's to regulate growth and cell death. And if they get mutated, we can have problems like that. Those are the most common mutations found. So here's a list of some of them, and it talks about, you know, what they normally do, like that, what category they're in, and then an example of one of the oncogenes, and this is RAS. And in us, there are 3 different RASes, so it's got a couple of different RASes listed there. But there's a set of those like that, okay? So we'll look at this, and I think I made it right here. So here's just a little table. I think these are the retroviral oncogenes that we'll learn about. Of the, say, 30, we'll just learn about these 5. And I ranked them by how much we'll learn about them. And they're all in our book, and they'll come up. Some of them we won't talk about till after the first exam. They'll come up over the next, you know, month and a half, 2 months, like that. So the first one is RAS, like that. And I'm just going to show you that this is that pathway that we're experts on that happens under a scab to help the skin heal. And the pathway is PDGF binds to the PDGF receptor, which activates RAS. And then RAS activates RAF and RAF and MAC and ERK and ELK. And then ELK is a transcription factor that's phosphorylated. So we're experts on that. And that's a normal, healthy pathway. And RAS plays a role on it. When there's a scab, under the scab in those skin cells, RAS is naturally activated. And it's activated in a way that we'll talk about, like that, okay? So it's on a normal pathway, yep?
For the exam, do you want us to know how, like, do you want us to draw this whole, like, sequence of events?
You'll see that you would just withdraw, you wouldn't even be asked. You'll be like, we've talked about that so many times, I'll just draw it next to my name on this exam. Yeah, it's something that you'll just get really, really familiar with. It's all about chapter 17. This is just one example. And like RAS plays roles in other pathways. But yeah, but to be more honest, Hannah, like there would be multiple questions on the exam about this pathway. Here's an example, okay? So I'll show you, and we'll talk about it more in chapter 17, but just to show you now that we're gonna learn more about RAS. And so here's this RAS, and it's a protein. And its role is on these signal transduction pathways, of which there are many, and RAS is just on some of them. There are many, many signal transduction pathways, so this is stuff that we'll learn about in chapter 17. So that's that RAS protein. And it's referred to as a G protein, and we call it a monomeric G protein. That just means there's just one protein molecule that makes up RAS. So it's a category of monomeric G protein. And what does that mean? That it's called a G protein because it's always bound to either GDP or GTP. So that's why it's called a G protein, because it's bound to GDP or GTP. When it's bound to GDP, it's inactive. And when it's bound to GTP, it's active. It gets activated by the receptor. So over here, there's going to be a receptor. The receptor is going to activate it when the receptor binds to its ligand, that signaling molecule. Small molecule that binds to protein is called a ligand. So we'll use that word a bunch. A ligand binds, it activates the receptor, it activates RAS. And to activate RAS, what happens is the GDP is released and GTP is bound, and they call that an exchange. So now, when it's activated, we have RAS bound to GTP, and it's active. And it's doing its job, and its job is to activate RAF, and it's just doing it like that, right? Until it gets inactivated. And it gets inactivated by breaking the GTP down by hydrolyzing it to GDP. So it's that RAS is also an enzyme, and the enzymatic activity, so you could make a little, like a mouth on that RAS, the enzyme activity is to take that GTP and to hydrolyze it to GDP and inactivate RAS. And that just happens, kind of randomly. It's on for a while and then it hydrolyzes GTP and it's off. Like that. So that's how it gets inactivated. Okay? So the scientists have mapped the mutations in RAS that cause cancer. All right? And the mutations could be either in the transcription unit that would affect the product, or the regulatory region that would affect where, when, and how much of it made. And all the mutations are in the transcription unit. And all of them are in the coding region. And all of them are point mutations that just change one amino acid. And all of those are in the region of the protein that is the enzymatic active site. So all the mutations that cause RAS to go out of control are in the enzymatic active site. And that means they decrease the activity. And that means they cause the GTP to be there longer before it gets broken down to GDP. So RAS stays active longer, like that, or it's active even when there isn't a signaling molecule, because it can't turn itself off. And it's very likely to come up with mutations. This is kind of an Achilles heel story, because normally a random mutation is going to inactivate something. And here it's inactivating the way this gets turned off, like that. So see, we're going to dive in a little bit on these in chapter 17 and beyond to say, oh, there you go. That's what's going on with that and why it's an oncogene. Right now, we don't have mutations. It's doing fine. It gets turned on and then it turns itself off. But it picks up mutations and it doesn't turn itself off very well, and we get uncontrolled cell growth because it stimulates that pathway. Okay? Yes?
Do you mind just explaining again why, like, what's happening that is abnormal for the RAS?
Yep. Yeah. So what's normal for the RAS is it's inactive. What's normal is it's always bound to either GDP or GTP. When it's bound to GDP, it's inactive. When it gets activated normally, it exchanges the GDP for GTP, and now it's active. And it stays active until it turns itself off by hydrolyzing the GTP to GDP. And the problem when it becomes oncogenic, cancer-causing, is it's picked up point mutations in the coding region so that it has wrong amino acids in the enzymatic active site, and it just can't turn itself off very well. It might turn itself off a little bit, it's not totally defective, but it just doesn't turn itself off well.
So it's
Yep.
not, like, able to hydrolyze the GTP in the same
Yeah, it
manner?
can't hydrolyze it. Yeah, very well, like that. So it just stays on longer than it's supposed to. Yep, like that. Yep, or yeah, yep.
I think I have two questions. My first one is, does this happen in the transcription step of the replication cycle?
Good question. The answer is no, because we started in the 1970s learning about RAS using that virus, and we found that virus could cause cancer because there was a host gene that it picked up. So then our attention leaves the virus. Who cares about that virus? We just learned in the 1970s it's the host gene. So then the guy goes from Wisconsin to Boston and he studies the mouse genes. Doesn't care about the virus anymore, ever again in his career, because the host cell genes could get mutated, like, too much sun at the beach, or, too many onion rings. They get mutated and come up with these problems, right? So that's a really important thing to understand that Godwin brought up, right? Is that the author, and it's very famous and talked about, that back in the day, they first found some of these genes that way. Then they left that virus story behind and they started working with mice, no viruses. And they found that mice that had cancer already had mutations in that RAS gene. Not to do with the virus, more to do with onion rings and sun rays. Yeah? Yeah, okay? All right, so you'll see, because in chapter 20, we're going to want to take that knowledge and stack one step on top of it, because there are drugs that now target these mutated genes, and they're for specific types of cancer. Right? RAS is mutated in a third of all cancers, but not all cancers. So we got to have a lot of different drugs because there are a lot of different genes that could be causing the trouble, like that, right? So we'll get to that. So we'll use this as our number one example, okay? So let me just flip back to this just to show you that, we're just picking some highlights, and you'll see that when we talk about that next step up in RAS, we'll use that one as the story and talk about a drug that affects that one, like that. And so, you could just write these down all now, but some of these are not going to come up till after the first exam, and we'll talk about them. I just want to show you, kind of put my arms around this list. From the 1970s, these are the 5 or so of the 30 that we'll be like, yeah, it's going to come up in our course, and a lot less, almost none. Like
Can
that.
you go back to the diagram of RAS from chapter 17? So the growth factor, is that the ligand, or is the ligand?
Yeah, the growth factor is the ligand, and a ligand just means a small molecule that binds to a protein that regulates it. So like hormones are ligands and neurotransmitters are ligands. It's a general biochemical term that the author uses some, I use some. The term we really like is it's generally a signaling molecule, like that. All right, so we're going to now do our in-class assignment. Yes, Joe?
I just have one quick question about RAS, and how you were saying it can be mutated in like some forms of cancer. How, like, I guess my question would be is like, how are they able to kind of like reset it? Because like, how you were saying it has like GTP to GDP, and then it can't hydrolyze GDP, so it gets like stuck there. Like, how are they able to like reset it so that like you go through the cancer treatment, etc. Like, how are they able to reset it in your body so that just doesn't happen again, like once you're like quote unquote like better?
Yeah, right. So the answer for RAS is there's no way to reset it. There's no way to get it normal again. There's no medicine that I'm aware of that regulates RAS. And the only way to get it better is to kill those cells with chemotherapy or radiotherapy, or cut them out in surgery.
Okay.
Yeah, so there's plenty of that, right? You just can't fix that cell,
Yeah.
right? Like that. The only hope is get it out of there, like get all those cells out of there, because it's mutated and we don't have a treatment for that one. Yep. And then Godwin, did you have one more?
Yeah, so my question was going to be, say we're taking an exam, right? And you just ask, what is the activation pathway of RAS? We should just be able to tell you that when ligand binds,
Yep.
GDP is active, and then when it goes through hydrolysis.
Exactly. Yep. Yeah, and that's a great question, right? Is draw a little picture of the cycle of activation and inactivation of RAS, and you just draw a little blob and say it's activated with exchanging GDP for GTP. It's active as long as it's bound to GTP, and it inactivates itself by catalyzing the hydrolysis of GTP to GDP, and we're back at the start. So it's kind of like a cycle of activation, inactivation. And you'd want to know that. Yep. All right, so we're going to do this homework assignment. It's going to be based on the clay model, and we're going to draw 3 pictures, and they're going to be eventually, you can tape together, but the paper's going to be up like this, in this orientation, 3 pictures. And you can come up, and I have some different colored pens you can use, like that, and get at least 3 pieces of paper. And you're welcome to work right next to somebody else. You have to do your own work, and at the end you'll either turn it in or take a picture of it and send it to me. But you can work right next to somebody else and just cheat off them the whole time.
What else do you do?
That's totally
We
legit.
need 3 pieces of paper?
Yeah, you're going to need 3 pieces. You might want to get a couple extras in case you need to start over. So just to get us started, and part of this goal is to review a little bit. Part of it is to add it to it and create a visual about moving forward, how we can take this to an advanced level. And then part of it is to encourage you to study like this, to build drawings like this and label them like that, and make flashcards and stuff like that. So these are the 3 pieces of paper just to show you, and you can eventually tape them together if you want, okay, like that. And then these 3 pieces of paper together, all together, they're going to represent one myoblast cell. All together, there are 3 all together. And in that, this is what it's going to look like. In that, this is going to, and you could even draw this on there right now. That's the nucleus. So our nucleus is what we're going to be focused on because it's going to be about gene expression. And the nucleus is going to be represented on all 3. So 1 cell, 1 nucleus covering all 3 pages, okay? And we're going to look at the expression of 3 genes. Does anybody know what the 3 genes are that we're going to look at? Yep, so the first one, the page on the far left, is going to be about expression of MyoD. And then what do you people think the center paper is going to be about?
Transcription.
It's going to be about another gene. What gene would it be probably?
Myogen.
Yep, so that central piece of paper is going to represent myogenin expression. And then what do people think the right piece is going to represent? It's going to represent the genes that myogenin activates. And what does myogenin activate? The right one is going to be muscle genes, because those videos and this drawing are about muscle differentiation. And so we have all the genes all the time, but it's not a muscle yet. And we're going to make it a muscle, and the way we're going to do that is we're going to activate expression of muscle genes like actin, myosin, actinomyosin, titin, tropomyosin, troponin, troponin T, stuff like that. So striated muscle fiber. 3 steps, 3 gene expression steps, 1 cell, the nucleus. So this is going to be a timeline. It's going to be time going from left to right. And it happens pretty fast, just in a couple of hours, that we go, oh, we're going to activate MyoD, then myogenin, then muscle genes. Okay, so let's just work on the left piece of paper, and we could draw a little gene here on the left piece of paper, and that could be the DNA, and it could be the DNA for the MyoD gene. And you have a lot more space than I do, but you can just draw that in. And I might have to cut out on this drawing and make a new one when we zoom in some, but I'll try to stick with this, okay? So you can label the parts of that gene, right? And so we're gonna have the transcription unit, and right next to it, we wanna have the promoter, and these are all parts of the DNA. And we want to have enhancer, like that. And you could label those to help you remember that every gene has those parts, like that. And then what's happening is it's going to be expressed. And so every cell has the MyoD DNA, MyoD gene DNA. We could find that everywhere. But we can't find the RNA everywhere. We can't find any MyoD RNA in early embryos because they're not ready to make muscles yet. We can never find MyoD RNA in the brain. The brain has the MyoD gene DNA, but it never has the RNA. So we want to represent that we are transcribing that transcription into MyoD RNA. We have lots of MyoD RNA. And anytime a gene is being expressed, the RNA is pretty unstable, and so it just has to be continuously made. So we're just continuously making RNA in the nucleus, right? This is just 1 cell in the nucleus, and we got RNA out here, okay? And then it's going to be translated, and so you can make a ribosome, out there and label it. I'm not labeling these, but that's a ribosome. And the ribosome is going to translate this into MyoD protein. And you want to make multiple molecules. So anytime a cell is expressing a transcription factor, it has lots of molecules of MyoD protein. And you can draw some of those in the cytoplasm of your center page, because we'll move over to that next step. Right, so the left page we're kind of done with, and it's labeling the MyoD gene and showing that it's expressed and translated to MyoD protein, and you have lots of molecules of MyoD protein. So anytime a gene is expressed, you can detect the RNA, and if it's a coding gene, you can detect the protein with, experiments like that in lab. You can grind the cells up, and you can detect the RNA or the protein. And that protein is everywhere in the cell like that, and it's in the cytoplasm, so you could just draw it in. It's all over here too, and you don't want to too much, right? But it's just one cell, right? So we made a bunch of it. And what happens is the MyoD has to go back into the nucleus, right? It gets translated and produced and created in the cytoplasm, but now in your center drawing, we got to go back into the nucleus, and it gets to do its function. And its function is to bind to the enhancer of the myogenin gene. Okay?
What's the
And-
gray dot?
Huh?
What's the gray dot?
The gray dot, or even all the black dots?
Gray dot outside of
The ribosome.
Oh, that's the ribosome.
Okay, thanks.
So the MyoD is going to go into the nucleus. It's got to get back inside the nucleus, and it's going to bind. Here's the MyoD protein, and it's bound to the enhancer of the myogenin gene. And so you should take time to color in. We got the myogenin gene, so it's got a transcription unit, and it's got a promoter. It's got this enhancer. And the key thing in the activation of myogenin is the MyoD gene binds. MyoD protein binds. And so I'm just going to put a little like a starburst around my MyoD protein, because that's a key concept. Is the transcription factor is activated, and it gets into the nucleus, and it does its job. And we're going to learn about that in chapter 9, regulation of transcription, and we've already learned about it some. Okay, so now we get to make the myogenin RNA. So we get a lot of myogenin RNA going like that, and that's the myogenin RNA, and it leaves the nucleus, and it's going to bind to a gray dot, ribosome, and it's going to be translated into myogenin protein. And that's going to be out in the cytoplasm, so you can make a lot of colored dots of myogenin protein. In the cytoplasm. Right? So we had our first transcription factor of MyoD, and it got made, it's in the cytoplasm, and then it gets in the nucleus, gets to do its job. Its job is to activate myogenin. There's a lot of MyoD. Most transcription factors have multiple target genes. We just are mentioning one, myogenin, but MyoD is binding to several genes and activating their expression. Myogenin is one of them. And so we get a bunch of myogenin made, and now on your right page, it goes into the nucleus. And it gets to do its job. And so over here on your third page, and I don't have much space, but you have bigger pages, you want to draw another gene, and it's a muscle gene. Let's just say it's myosin. Myosin and actin are, the central players in the striated muscle fiber. And so the myogenin binds to the enhancer of the myosin gene, and we got the different parts of the myosin gene, and it's going to activate transcription of the myosin gene, and we're going to get that out in the cytoplasm, and the gray ball is going to translate that into a protein. And I'll let you catch up, because that's the basic drawings we're doing. So I'll let you catch up with that and ask questions about, just how to roll through that. It's 3 genes. The story is really similar. Each of the genes looks really similar. They just have a different label, name, MyoD, myogenin, and we'll say myosin, a muscle gene. Then MyoD protein gets into the cytoplasm and activates myogenin, and also other genes, because there's lots of molecules of MyoD. Then myogenin gets expressed and translated as a protein in the cytoplasm. It gets back in the nucleus and binds to the muscle genes, myosin and other muscle genes, and activates their expression. So we have a pathway to gene expression, okay? Okay? All right, and so we're done with the basic drawing, and now is when we're going to take a look at this and see something. So what's going on here are some feedback loops. Okay, so I'm just going to draw over mine, and I'm just going to draw over my whole thing, and you don't have to mess yours up like this. But see, here's a feedback loop, right? This is DNA, it's storage information, and some of it gets output. It's like the output expression, and then it feeds back. It feeds back on the storage information. So this gene expression, because it's a transcription factor that's being made, creates this output loop like that. Then the same thing happens again, I'll make a little different color, where the myogenin, that's stored DNA information, and its output, we're making an output, but since it's a transcription factor, it also is a feedback loop. We're regulating the stored DNA information through this output loop. And in this example, and we just have 2 minutes left, well, we have 5 minutes left in our class, so we're doing great. So this is it's an important example, but it provides a pretty simple example, because in this case, the feedback loop is the MyoD transcription factor gets made, and it does its job. And then the myogenin gets made, and it does its job. Okay? So now we're just done drawing, and now just think about this. Every transcription factor has that story. The transcription factor is an output-input feedback loop. We've got the stored DNA that's pretty stable and steady throughout our lives, and we're going to express a gene. And if it's a transcription factor, that transcription factor is going to turn around and affect more output. Okay? And then the last thing is you can make, and again, you don't have to put these big feedback arrows on, but if you have, you can make a little starburst around this one, because that's a step where that could be regulated. So we can regulate that output-input pathway. What's an example of that? Well, elk has to be phosphorylated. So on this pathway with the muscle, it's kind of like dominoes falling. But if it's another example like elk, it's like the dominoes fall, but then the elk domino home, it only falls part way. But it needs to be phosphorylated before it really falls. Or FOXO needs to be dephosphorylated, needs to be allowed in the nucleus. So in these loops, we have different transcription factors that can be regulated. Here's a really good one. In our bodies as humans, we have genes that are off in our childhood and get turned on by estrogen or testosterone during puberty. The receptor for estrogen and testosterone is our transcription factors. So here's the estrogen receptor getting made in our childhood and waiting to complete the feedback loop. And so a huge part of the complexity of the storage of DNA is how it has the stored information to create these feedback loops that regulate the timing of the output. And that's why we're building that list of transcription factors and how they're regulated. MyoD, it's just whether it gets made. Myogenin, whether it gets made. But then FOXO, whether it gets phosphorylated. ELK, whether it gets phosphorylated. And we'll have more examples of that. So, see, we just did the clay model video and reviewed and learned that better, but we also use that now to step back and see, oh my gosh, this is the huge central story of life on our planet, is that every cell has DNA as the storage molecule, and in the storage information, it includes information to create these output-input feedback loops that include regulation by the cell or the organism or even the environment. In some species, reproductive maturation and the production of estrogen and testosterone are regulated by the environment. So those are feedback loops that affect the input-output regulation of DNA expression by having other loops of feedback in the organism and in the environment, like that. So this is a big picture thing that we want to just have in our head of where we're at and why would we spend so much time talking about these transcription factors. And it's because they're a big, big show on this stored information and how we output it during our lives to make our bodies and live our lives. Okay, good. So you can either take a picture like that, or you can write your name on it and hand it in. I brought tape, if you want to tape yours together, just to, it's another learning thing, right? It's one cell. We made 3 parts of one cell. So we intentionally did all that stuff, and then, so either take a picture and send it to me like that, just boom, or just hand it in, with your name on it, and then we'll just clean up and we'll have nap time. Yes! You guys earned